Hi everyone, my name is Dr. Sarah Zukoff, and I am the new entomologist at the Strawberry Research Center at Cal Poly. I am coming to you from Kansas State University right now where I am, but in about 2 weeks I will be at Cal Poly. And right now you can see the corn behind me. I am actually in a Kansas cornfield, and I'll be doing this presentation from Kansas. But very soon I'll be there in person, and I hope to meet you all. Thank you.
Well, I'd like to tell you a little bit about my background. First, I'm from South Georgia, and I received my bachelor's and my master's degree from Georgia Southern University, where I worked on spiders, ticks, and sandflies. While there, I concurrently worked at the University of Georgia on several crops as a crop consultant. Then, I went to the University of Missouri, where I got my PhD and worked on corn under the USDA ARS Plant Genetics Unit. From there, I went to Kansas State University and worked as an associate professor in field crop entomology. And from there, went to California. Now I'll be at the Cal Poly Strawberry Research Center. Well, that's enough about me. Now on to the research I wanted to present today. This work was conducted last year by Peter Scherer, my predecessor, and our entomology technician, Jose Alvarado Rojas. This work looked at the quality of commercially produced predatory mites that are used for spider mite control. Throughout the many crops I've worked on, spider mites have been of particular interest to me, and I've studied them for many years. I'm looking forward to working on spider mites now, and with the farmers who work in strawberries. Since two-spotted spider mites are important pests of strawberries, I wanted to mention a little bit about them and the damage that they can inflict. These mites have a very fast reproductive rate where they can lay lots of eggs and under warm temperatures can complete their life cycle in as little as 1 week. All that feeding and reproducing quickly dries the plant out, which can lead to a reduction in strawberry yield. They usually can cause stippling, scarring, and bronzing of the leaves. Here's a nice video that Jose took. This is up close on a leaf. You can really see where their feeding damage has occurred. All those little stippling and spotted areas. One of the ways that farmers manage spider mites is by using predatory mites. These good guy mites can be purchased from commercial sources and are usually either one of two species. One of them is Phytocelius persimilis and the other one is Neocelius californicus. These and other species of predatory mites sometimes occur naturally and in some areas have already been established. Here's another nice video by Jose. You can see a predatory mite feeding on a two-spotted spider mite here. The predatory mite is orange and shiny and resembles a classic piece of hard candy, while the spider mite is smaller and has a softer body that isn't shiny. Now the predatory mite latches on with its fang-like mouthparts and will continue to feed on the spider mite until it's basically dry. It will leave the discarded shriveled husk and go search for another as soon as it's done with this one, which is why they make such good predators. Here you can see the strawberry covered in pest mites and their webbing. Now, since these spider mites can cause a lot of damage in strawberries, farmers will order the predatory mites and place them in the strawberry fields with the expectation that the predators will clean up the spider mites. These mites can work in tandem with some chemicals or by themselves depending on the farmer's practice. However, what the farmers were seeing were lots of areas that the spider mites would just keep reproducing and causing damage. So what's going on? What's happening with these predators? So Peter and Jose decided to investigate the quality of the predatory mites that the farmers were purchasing. So I will be presenting their results. This work was funded by the California Strawberry Commission. The specific objectives of this study were to assess the abundance within the containers and to see if it was accurately depicted on the label. To also see if the females were reproducing and actually producing eggs, and to see if they survived and if their babies survived, and if the mites actually appeared healthy. For this work, 3 standard quality parameters were used. For both species, the amount of mites in the shipped container must exceed the amount listed on the label. The second parameter was that 80% of the predatory mites must be alive in each container. The third one is that the predatory mites must lay a certain number of eggs per female, and this is to ensure that they will continue reproducing in the field after they are released. Now, in these strawberry fields, healthy predatory mites should have no trouble continuing to live happily and reproducing. If all these 3 parameters are met, then a container would be considered acceptable quality and considered worth the money. If not, it is unacceptable. They categorized predatory mites as either gravid or pregnant females, or males and immatures, so those that are reproducing versus those that are not. The chunks of debris in these pictures are vermiculite and are the substrate used to house and ship the predatory mites. The small red dots in the circles are the predatory mites. To do this work, you need to quickly identify each life stage of the mite by sight, which is kind of difficult. A phytocelius mite can consume around 7 adult spider mites in one day, or 20 immature spider mites in one day, or 25 spider mite eggs in one day. And generally, there are around 4 females for every 1 male you find in a healthy population. Now, the females are generally larger and darker than the males. You can see the male at the bottom, it's smaller and lighter colored than the female. So in addition to counting the predatory mites in each shipment, they also started noticing stressed mites. So what does a stressed mite look like, you might ask yourself? Well, they look discolored, can be white, cream, or even darker than normal color. These mites will often have a lower egg-laying rate and will not eat as much as a healthy predatory mite. They also might have a large rectal plug, which unfortunately gets stuck on the leaves and they could die. So here you can see the adult predatory mite, and here is the predatory mite egg, which is oval shaped, and here is the spider mite egg, which is circular. And you can see this predatory mite actually starting to feed on that spider mite egg. So what did they find? Well, let's look at the predatory mite Phytocelius persimilis first. Recall that the first quality parameter was that the abundance of the predatory mites within the container must exceed what was on the label. Now here you can see the 25 containers of commercial predatory mites they received, and unfortunately only 3 of these actually met this quality parameter. The next parameter was that 80% of the predatory mites must be alive in the samples in order for it to be considered a quality sample. So of these 25 that they received, they were only able to measure the survivalship in 17 of the containers, but out of those 17, only 2 actually met this quality parameter. So the third quality parameter was that each female had to lay at least 10 eggs per female. Now, of the 25 that were received, again, only 17 they were able to look at fecundity, or the amount of eggs that were laid per female. And you can see everything that's above the yellow line, there's only 10 of them. So 10 of the 17 that they were able to measure actually met this quality parameter. Now let's switch to Neocelis californicus and look at what they found for those mites. So here, they looked at the quality parameter for the abundance. So did the abundance within the container meet or exceed what was actually written on the label? And so if you look at these, they were only able to receive 7 of these containers, but 4 of them actually met this parameter. So of the containers they received, they were able to measure survivalship on 3 of these containers. And so 1 of the 3 actually met the 80% or more survival per container. So that's kind of low. And the third parameter was to measure fecundity, or the amount of eggs laid per female. And for this species, they were supposed to be laying 7 eggs or more per female. and what they found was 2 of the 3 containers actually met this parameter. So in summary, only 1 container out of the 32 bottles met all quality parameters, and less than a quarter of the containers met their labeled abundance. So there's more work to be done with this. So I would like to thank Peter and Jose for conducting this assessment, and I would like to thank the Strawberry Commission for funding this work. I hope to pick up where they left off and continue to look at predatory mites and problems associated with their release in strawberry fields.